Good evening, everyone. Whether you're joining us remotely or here in the audience tonight, we welcome you to our annual Jefferson Pilot Lecture featuring the 2020 Jefferson Pilot Professorship Awardee, geologist Dr. Jeffrey Kuglitsch. The Jefferson Pilot Professorship is an endowed award established in 1981 through the generosity of the Jefferson Pilot Life Insurance Company, the highest and most singular honor North Carolina Wesleyan College bestows upon a member of its faculty. The honoree is nominated by a committee of faculty and appointed by the president. Those eligible to receive this award are tenured members of the faculty who have clearly distinguished themselves in ways that set high standards of excellence to which the whole community can aspire. As teachers, they demonstrate a wholehearted dedication and rigorous professionalism in forwarding of their discipline, contribute to new understanding and knowledge, and demonstrate a lively commitment to lifelong liberal learning for themselves as well as their students. As members of the Wesleyan community, they give unstintingly to the aims in the programs of the college, both with their times and means. Dr. Kuglitsch joined Wesleyan's faculty in the fall of 2006 after a successful career in geological sciences, working as a lecturer and practitioner in the field in Wisconsin. He earned an MSc in geology from the University of Wisconsin-Milwaukee and a PhD in geology from the University of Wisconsin-Madison. Beginning as an attorney by trade, Dr. Kuglitsch also earned a BA in history and political science from the University of Wisconsin-Milwaukee and a JD from Washington University School of Law in St. Louis. Dr. Kuglitsch is a member of numerous academic and professional societies, including the American Association for the Advancement of Science, the Geological Society of America, the Paleontological Society, and the Society for Sedimentary Geology. He also reviews literature for the Geological Society of America and Lithia. While at North Carolina Wesleyan, he has taught courses in physical, planetary, and marine geology, as well as special topics courses in marine micropaleontology and fossil fuels, mineral resources in the environment, and his contributions to the college have been numerous, including serving on college-wide committees and task forces while also developing relationships with public and private schools, giving presentations and workshops to K through 12 students. Please join me in welcoming Dr. Kuglitsch to give his talk entitled, The Ancestor of Dragons, a Riddle Wrapped in a Mystery Inside an Enigma. Dr. Kuglitsch.
Well, my predecessor, Dr. Lee Templeton, was here back in March 28th of 2019 doing his presentation. I'll come to that in a minute. I was supposed to have done mine a year after his, and it got postponed. I should suggest that was intentional on my part because I'm enjoying being the warm-up for the presidential debates this evening. I don't know how many of you are staying, but the political science department here is doing that presentation live at 9 o'clock. But I admit it was just dumb luck on my part. Now, when I was announced as Jefferson Pilot lecturer for this year, I decided it might be nice to have some sort of connection, some kind of continuity between my talk and Dr. Templeton's. And I decided I would do that with the title. So this is the original poster I was going to hang up back in March of this year, called The Ancestor of Dragons. And the subtitle there, A Riddle Wrapped in a Mystery Inside of an Enigma, we'll get to a little later, but I suspect many of you recognize that as a quote from Winston Churchill. Before we can talk about ancestors of dragons, we have to think a little bit about what a dragon actually is. Some dragons are purely imaginative. This is the Kaskaskia dragon, located originally in Kaskaskia, Illinois, in the southwestern part of the state, not too far from St. Louis and the Mississippi. It's since been moved to Vandalia, not too far away from Kaskaskia. And you can, if you go visit it, go to a local liquor store and purchase tokens and put them in a slot next to the dragon and make it breathe fire. You might also notice it has its antlers wrapped up in some sort of stockings up there. It was a cold season, otherwise it's seen in a top hat. This is a purely imaginative dragon. Other dragons will have some foot in reality. What you're looking at here is an etching from the 1880s. It's an engraving of a dragon statue in Klagenfurt in southern Austria, in the province of Carinthia. And the story was in the 1200s that this creature was harassing travelers, consuming virgins. Apparently it was very discriminating in its dinner tastes. And two young men, well, it depends on which virgin you read. It's either one young man or two young brave individuals decided they were going to do this thing in. And the story is either that they hooked a bull up to a big chain and fished for it. Apparently it hung around in the river a lot of the time, and pulled it in like a big fish and dispatched it, or they stuck the bull on some kind of a platform and the bull dueled with it. At any rate, in 1860, a statue was made of the dragon. And the dragon actually, from the neck up, is based on reality. In the 1300s, a skull was pulled out of a small local quarry, and that became the model for the head of this dragon. Now, from the neck down, it's all pretty imaginative, but you can tell it has a reasonably dragon-like head. The statue there is of Hercules. I don't have any idea why that is. It should have been one of the guy that dispatched the dragon. At any rate, that skull was based on the skull of a woolly rhinoceros, a late Ice Age mammal. And the skull that the dragon is based on, and you can actually go see the skull in the museum in Klagenfurt, lack the horns. Very often the horns get separated from the skull in this particular species. And so this dragon actually has some factual inspiration to it. And I became interested in that, particularly after reading this book by Adrian Mayer, The First Fossil Hunters. Mayer is a classical trained folklorist. If I'm recollecting correctly, she had got her PhD at Yale. It was one of the Ivy League schools in any event. And she became very interested in how ancient cultures treated fossil remains, particularly vertebrate remains. And, you know, she wanted to explain a lot of mythological interpretations. And I have a vague recollection as a child of being home with the flu. And I think my mother had run out of books to bring me to read, and I recall at some point she brought me a volume of Norse mythology and Greek mythology, and maybe the Odyssey was tossed in there as well. And I remember reading all these stories and finding them quite intriguing, but you know, you're reading about guys getting chained to rocks and giant eagles coming and eating their intestines every day, and they grow back and do it all over again, you know, thinking back to the Andromeda myths, you know, and one-eyed giants. And I guess nowadays, reading those, I would have wondered what those guys were smoking. Back then, I just thought, you know, those are some pretty weird stories. But Mayer's work, you know, made me think a lot about this stuff, and particularly one example, and I'm going to liberally expand the term dragon now to include a wide range of what we historically have regarded as mythical beasts. She examined work by a group called the Scythians, who were around between 900 BC and 200 BC. They basically they were nomads. They inhabited the area between Asia Minor, what we would modern day Turkey, and then all the way over into western China. And this area in western China, you can see the Gobi Desert there and the Altai Mountains. The Scythians had gold mines in those areas. They were very accomplished metal workers. And that area, the Gobi Desert, is an area where there happened to be a lot of exposures of vertebrate fossils of a lion-sized dinosaur called Protoceratops. In any event, the Scythians did just marvelous gold work. And looking at this rather ornate piece, one of the interesting things people have found is that they tended to be very representational in their art. Representational to the point where botanists can look at plants in their designs and literally identify them frequently down to species. But there are a few minor exceptions to this representational approach. And one of them relates to the lower part of this neck piece that we're looking at, which I've blown up here. You can see a horse in the process of being consumed by two rather nasty looking animals, four-legged, apparently predators with wings. And if we could see a little closer, you would see that they were beaked. Some of you may have already recognized these as the mythical griffins, the flying lion-sized predators, with a raptor's head. These are very much the exception in Scythian artwork, something so fantastic. Well, Mayer, was familiar with the geology in the area where the gold mines were, and she looked at these fossils, reconstructions of this lion-sized dinosaur called Protoceratops, and you can see looking at this thing, it has a nice beak-like sort of skull. It's, again, about the size of a lion. It's a quadruped. This is just a modern reconstruction of what it might look like, probably a little more decorative than the actual animal. And she saw this as being the model for the griffin. Her belief was in that, all likelihood, the Scythians were looking at fossil remains of Protoceratops, and they were doing a paleontological reconstruction. Well before the Klagenfurt dragon. Historically, people regard the Klagenfurt dragon that we looked at as the first paleontological three-dimensional representation of a prehistoric animal, albeit 90% imaginative, but they got the head right. The griffin, again, it's about the right size, it's a quadruped, and the Scythian interpretation of the remains is that you've got an animal with a large raptorial beak. The frill on top of the skeleton here generally isn't preserved, so most of the skulls don't have that. And the other thing she noted is that this animal has a very long shoulder blade. And the Scythians, having been very acute observers of nature, undoubtedly had seen the skeletal remains of raptorial birds who have substantial long shoulder blades for the attachment of wings. So the idea simply is that they saw these remains and it was only natural to interpret them as some sort of large winged raptorial animal. Beyond that, many of the representations, and this is one of her drawings from a bronze relief work, show the large parent animal sitting above young animals in almost a nest-like setting. And the Gobi Desert is a place where dinosaur eggs were first brought to the attention of the Western world. This is a picture of a clutch of dinosaur eggs that were brought by Roy Chapman to the New York American Museum of History. It was quite a to-do when it happened. And Mayer speculates that the Scythians probably saw dinosaur eggs, Protoceratops eggs. It's clear that these animals, we now know, did basically arrange their eggs in a nest-like arrangement. So the idea that these were interpreted as being some sort of winged beasts, really, that has a very substantial foot in the real world. Now, they may have gotten the wings wrong, but given the circumstances, it was a very natural mistake to have made. She also speculated that the Scythians liked to use this stuff in their ornamentation simply because it was a good way to discourage people from encroaching in their gold mining areas. The thought was that if people thought these areas were inhabited by these nasty griffins, they might not want to nose around looking for gold. So there may have been an ulterior economic motive. Other examples that Mayer cites relate to an area in the foothills of India referred to as the Sweelock Hills, that have a lot of Miocene vertebrate remains. These are relatively recent in terms of geologic history. What you're looking at here is a reconstruction of a huge elephant-sized giraffe with gigantic, in effect, antlers on top. Normally, these skeletons are found disarticulated, so more often than not, people would find a single skull. This is one hanging in a museum in Tübingen, and you can see it. I mean, it's got a very dragon-like kind of appearance. And the locals in India basically had all sorts of lore about dragons in these areas. Here's another giraffe species called Giraffopteryx. Again, you can see these multiple horns up on the front and a very, dragon-like potentially sort of skull. She cites a traveler named Apollonius, who came from Asia Minor, who traveled through the area, and he described seeing fossils up in the high foothills, and he said that the ones up in the high foothills were generally larger than the remains that he saw in the lowlands. They generally had deep eye sockets, prominent brows, and nasty-looking horns. He also recorded the locals as saying that sometimes the earth shook when these animals burrowed into the earth, which probably was inspired by the regularity of earthquakes that occurred in that area. So one of the things I found kind of refreshing about having read that book is I think we can very much look at a lot of mythological and folklore accounts of phenomena, and in reality, we tend to have a lot of hubris and tend to dismiss these things, but in reality, a lot of them had a connection to underlying reality. So based on what we've seen here, I would argue that at the very least we can conclude that dinosaurs were some kind of vertebrate organisms, not unlike ourselves. And the organisms I work with, I think, are very close to the line, to the original common ancestor that vertebrates shared with a lot of other organisms. This is my marine kind of dragon, and you see this guy with these big conical mouth parts here. A group of Vikings in a longboat would unless they were in a really small longboat and they were 1-inch tall Vikings, they wouldn't have too much to fear from these guys. The little mouth parts you're looking at, their range in size. Basically, think of a large piece of dandruff to a very small piece of dandruff. These are phosphatic remains from an extinct animal called conodont. The hard parts are roughly made out of the same material as your teeth, calcium phosphate or apatite. And normally, they're found like this. They're found as isolated elements historically. These are just some pictures from my dissertation material. And they come in a variety of forms, these simple cone-like ones and other forms. They have a long range. This is a geologic time scale that I have cribbed from one of the textbooks I use simply because it's nice and graphic. If we're looking for the ancestor dragons, we don't have to worry about most of geologic time. The solar system basically formed about 4.6 billion years ago at the bottom of, at the bottom there. And it wasn't until about 3.9 billion years ago, 3.8 billion years ago, that the Earth had a relatively stable, solid surface and started accumulating water and forming oceans. But the reality is, for most of the time from 3.8 billion years ago, all the way up to a little short of 600 million years ago, most of what was living in the oceans was single-celled organisms. And their fossil record, you got to stare there a lot. You have to stare pretty hard to see anything unique. I mean, there are a lot of circular impressions, chains of circular spherical objects, all single-celled. The time period above that you see expanded on the right side is sometimes called the Phanerozoic, meaning era of abundant life. So roughly from 600 million years ago to the present day. That's called the era of abundant life simply because what it really is, it's abundant multicellular life, and multicellular life that has a lot of hard parts. We don't get a good fossil record until we have animals with larger animals with hard parts to preserve. So this part of the scale is very finely divided into these series of geologic periods you see here, starting with the Cambrian and going all the way up to modern time, which you can call the Quaternary. There's some other names we use as well. There's debate about now whether we're in a new period called the Anthropocene, simply because humans have had such a major impact on the planet. This scale was developed primarily using relative dating. That is to say, what people did was they studied the distribution of fossils within the rocks, and they found that some fossils were always found lower in a sequence of rocks than others, and they worked these things out on a real crude term. If you found dinosaur remains in a unit of rock, you could conclude that you were in the Mesozoic time, right up there. Oops. I should have made a bigger cursor here. If you look in the middle, that green area there, starting about 248 million years ago, going to around 66.5 million years ago, is a Mesozoic. You find dinosaur fossils, you know you've got to be in that time span. Now, that's not very great resolution. It doesn't tell you a heck of a lot. But basically, this time scale is worked out in relative terms. Later on, all the numbers there on the side got put in doing what we call radiometric dating, based on the breakdown of radioactive elements that gradually turned into stable daughter products. And that's all I'm going to say about that, because I have a lot of material I want to cover. At any event, that time scale was basically worked out using fossils. Now, when we look for fossils, we're basically talking about sedimentary rocks, and this is another set of slides from the textbook here. We're looking at a picture of the Grand Canyon, and you can see these nice thick layers of rock. We're looking back toward the back of the canyon. The study of rock units is sometimes referred to as stratigraphy, and it focuses primarily on sedimentary rocks, but it really covers all of them. You may remember your basic rock groups. Igneous rocks all start off as molten material and then solidify. Sedimentary rocks we think of as being made of bits and pieces of other rocks that have been broken down through a series of weathering processes and then glued back together to make a new rock. And the third group you may remember is metamorphic rocks. You take an existing rock of any kind, you squeeze it, you heat it, short of melting it, and it'll change into a new form of rock called metamorphic. We're going to worry about sedimentary rocks in here. The true study of these sedimentary rocks, from a scientific point of view, originated with the work of a Danish physician named Niels Steno. He was also a scientist in his day. He lived from 1638 to 1686. And he formulated the basic rules we use to look at sedimentary rocks. One of the things he determined right away is that sedimentary rock units, they extend in a horizontal position. They're generally laid down horizontally. That's referred to as the principle of original horizontality. They extend indefinitely until they bump into some other kind of rock or they gradually thin out. He called that lateral continuity. And then the really big one is something called the principle of superposition, which to all of us seems pretty straightforward, but in his day and age was a radically new approach to looking at sedimentary rocks. He said, look, the rocks on the bottom of a group of sedimentary rocks, in the absence of something weird happening to them, the ones on the bottom are going to be older than the ones on the top. That's the principle of superposition. And that led him to the conclusion that there's a history there recorded in these rocks. Obviously, sediment falls through the air or it falls through the ocean and piles up from the bottom up. Yeah, it makes sense the stuff on the bottom is going to be older than the stuff on the top. And so geologically, if we look at the Grand Canyon, we break it up into a number of units that people call formations. Those formations typically, and you see on the right side of the diagram here, they're typically named after a place where they're easy to study and after the rock type. So these are what we call lithologic formations. Lithology is just a fancy name for rock. So you see the Coconino sandstone in the middle there, and up on the top you see the Kaibab limestone. And generally formations are all deposited in the same kind of environment. And the thing you have to keep in mind is that environments migrate. Think about, well, for a minute we'll make believe the Coconino sandstone is a beach sand. Some of it is, but most of it is other back beach dune deposits and river deposits. But think of it as a beach sand for the moment. If the ocean starts to rise, what's going to happen? The shoreline is going to creep inland, and it's going to cover the continent. And as long as it keeps rising, it's going to keep depositing a deposit of sand behind it. And that's how you can get a beach deposit to cover a very wide area laterally. This is where we get this principle of lateral continuity. Now, one of the things you want to think about is the sand that was deposited when the ocean was low is going to be a little bit older than the sand that's deposited once the ocean rises. But for the most part, we tend to ignore a lot of these changes. In general, the Coconino sandstone everywhere in the Grand Canyon area is going to be about the same age. The other thing that stratigraphers like to do is they like to correlate rocks. They like to be able to say the rock in this area is the same age as the rock over here. Now, when you're looking at the Grand Canyon, it's pretty easy to stand on that canyon and look at the Coconino sandstone in the foreground and look way across, and you can see that same thick white unit on the other side of the canyon, and it doesn't take too much thought to realize that the two were connected, and that was a continuous unit of rock. But it gets a little more complicated when you're correlating rock exposures 100 miles apart or maybe a few thousand miles apart. When we're doing Earth history, we want to be able to say some of the rocks in the Grand Canyon are the same age as maybe some rocks in Britain or in Africa. And to do that, one of the things we tend to rely on, again, is fossil content. My specialty, which is really kind of old-fashioned these days, is what's called biostratigraphy. Bio for organisms, you're just looking at the distribution of fossils within the rocks. And most paleontologists that become biostratigraphers will specialize in only one kind of organism, maybe two, and they will only deal with a narrow part of geologic time just because there's so many different species of any given group that it takes forever, to be able to learn and recognize them. So I guess in many ways we're rather boring specialists. So what you're looking at here is a diagram of the distribution of some conodont elements. This is from work of some guys named Barrick and Clapper. This is a couple decades old. And the line there that you see reflects the development of conodont elements from an organism it's a genus called Cocolella. And Cocolella ranuliformis, the line at the base of the diagram here, extends quite a ways up. We're going from older geologic time to younger geologic time. And this is from a period of time called the Silurian, just ballpark of a little over 430 some million years old. Couple things to note. At the bottom, you see something called the Amorphinothoides zone. The Amorphinothoides zone stops at that line. I'm only mentioning this now because we're going to come back to this guy. This guy is called Pterospathodus amorphinothoides. It's actually got a subspecies. Above that line, you don't find it in the rocks. Any rocks younger than that, it's absent. Because what we're doing essentially is using the ranges of these organisms to constrain their age of rocks. Geologically, species originate in relatively short periods of time and spread out throughout the earth. Their extinction from a geologic point of view is equally almost instantaneous. And so we can use their origination and extinction points to define discrete units of time. And the other thing I want to look at here is you can see a line labeled Walliseri. Cocolella walliseri is a descendant. It branches off from Cocolella granuliformis. So basically, the work that I've concentrated on, and I admit I guess I have a high capacity for rather dull work at times. Originally, when I started working on my thesis and later my dissertation, I was endeavoring to correlate rocks in southeastern Wisconsin with those up in northeastern Wisconsin. You can see the Door Peninsula up here, and then in upper Michigan up here, and then also in the Michigan Basin. The Michigan Basin area, those little dots there all represent material from cores. 400-some million years ago, this area was a basin. The center of that basin in Michigan was slowly subsiding, and it kept filling up with sediment. And that subsidence continued, and eventually it got buried. We're going to be looking at some cores from 4,000 feet or so below the surface. At any rate, it's very difficult to correlate rocks in southeastern Wisconsin with those up in the Door Peninsula or up in upper Michigan for a couple of reasons. Number one, there's not a lot of exposure of rock. Most of the rock in that area is covered with a thick veneer of glacial debris. So if you want to actually examine the Silurian age rocks, and these are all what we would call limestone and dolostone, what we call carbonates. They're all rocks that were deposited in shallow tropical seas at a time when North America was sitting a bit south of the equator. We'll come back to that a little later. So this is a large operating quarry in southeastern Wisconsin in which I basically got the sample from the top to the bottom looking for the conodonts that I work with. I got to do that there simply because I did educational tours for school children at that quarry as a public relations effort. They wouldn't have let me in otherwise. So I had, got fortunate. Working quarries, people worry about letting you in simply because, you never know when OSHA's going to show up and they see you walking around in the quarry, you can end up getting in trouble. The other places you can find rock to work with is in abandoned quarries. This is an abandoned quarry in upper Michigan. You can probably see the fellow up there about 3/4 of the way up the screen. That's a graduate student from Western Michigan University at Kalamazoo named Muhammad Al-Masawi. He and I are working on a couple projects together. And let me tell you that I was not taking undue advantage of him after snapping that picture. I scrambled up the rock face and helped him collect. In another place, sometimes you can find natural exposures of rock, but they're usually not very extensive. So what you're looking at here is the exposure of a small Silurian reef mound. Some of my running buddies in the audience might recognize my able field assistant, my wife Mitzi, standing back there. And I'd been looking for this outcrop for years, and it's another story, but she actually helped me find it. I will say I was tempted to put another slide in here after this that would have showed her snoozing away laying on the rocks, but I didn't want to live with the consequences of that when I got home tonight. So we'll leave that with no graphic representation. Now, Mohammed and I, the prior slide, you saw him up on the cliff. What we do is we take a sample, then we measure our way up a meter at a time, take samples at 1 meter intervals, and then I take this stuff back to the lab. These are actually some of the samples from that particular outcrop. The other thing we work with are samples from cores. This is a core facility. That is an anonymized Wesleyan student who a few years back went and helped sample cores. This is from a big facility in Kalamazoo called the Michigan Repository for Geological Research and Education. So here's a piece of core sitting in the lab. And what I do with these rocks is really, I guess, kind of nasty. I take the core sample or the whole rock, I take roughly 1 kilogram of it, and I suspend it in a net. you can't see the net real well there. It's basically made out of plastic rain gutter guard, and there are some plastic chains from Lowe's. I suspend it in a 5-gallon bucket from Lowe's that's full of dilute acetic acid, basically really strong vinegar. And limestone, which is made out of a mineral called calcite, the calcite will dissolve fairly readily in that vinegar. This is a picture of the lab fairly early on, where I don't have too many buckets going. I later rearranged things, and I've had as many as 104 buckets cooking in there at a time, which is a tremendous amount. Some of you may have been up in the second floor gravely and occasionally smelled vinegar upstairs. At least I've been told once in a while it smells like a pickle factory. I've been smelling it so long I no longer notice it, except when I mix up fresh acid. After the sample sits for a few weeks, I take it out of the bucket, I set it on a pair of nested sieves, and I rinse the pieces off. Then they go back in the bucket. It takes a long time to digest these rocks. The sieves are similar to what you played with when you went to the beach as a kid. Different widths on the wires in these things. The one on the bottom collects material that's really fine, basically small bits of clay that aren't going to have anything I'm interested in. That gets all tossed because I don't want to waste time putting it back in the acid. The top one catches large chunks that aren't going to have freed conodonts, and that stuff goes back in the bucket. And then the conodonts are found in that middle sieve. And so this stuff is rinsed through these nested sets of sieves. I reuse the acid. Periodically, I gotta use a new fresh batch. The stuff gets rinsed, and then I end up with beakers full of residue. And the residue will be little bits of undigested calcite or dolomite and other odds and ends, including a small number of conodonts. And these are 250 milliliter beakers. Some of my samples will yield well over, in terms of volume, will fill 1,000 milliliter beaker and sometimes multiple 1,000 milliliter beakers. And so what I need to do in order to find the few conodonts that are in there, I use what are called separation funnels. These funnels you're looking at are full of a heavy liquid. It is a liquid that is so dense. It's dense, but it's not sticky. It's very low in viscosity. And what happens is I put roughly 50 milliliters volume of sample in each of these sieves. I can do 6 at a time. And I stir that stuff around periodically, and then I let it settle. Here's one that's just been stirred. You can see all the dark sediment. And if you look at the bottom where that little stopcock is, you can see some dark residue. Here's a close-up. That's the heavy material. In that heavy material will be little bits of iron pyrite and hopefully some conodont elements. The conodonts are just dense enough that they will sink to the bottom, unlike the calcite and bits of quartz and other odds and ends. So these are a bunch of heavy residues that I've run through the funnels after I've opened up the stopcock. I then stop it before all the light stuff comes through. And here you can see this is a bunch of leftover light material that hopefully is free of any conodonts. I will always scan that. If I see something interesting, I might save it, but most of that stuff gets tossed. Currently, a lot of it has ended up filling in a depression at the end of the driveway of my wife's house up in Durham. So it's odd. It's kind of odd. She's got sediment from Wisconsin sitting at the end of her driveway. What I do then with that heavy residue, after it's been dried in an oven, I spread it out on a tray like this, and you can see it's divided up into squares. And I'll take an artist's paintbrush, and this tray will go under a binocular microscope, and I will move it from square to square. These are actually some coarse particles I put in here just so you could see them. And every now and then I will find a conodont, and I will dip the brush in a little bit of water, give it a fine point, and pull it out and set it into a slide. Otherwise, it'd be impossible to find these things if I didn't have some way of separating, in effect, the conodonts from the rest of the chaff there. Now, the process I've been describing was something that was developed in the 1930s. That's when people discovered that you could actually you could actually liberate these things using acid. Before that, they were mostly studied in split pieces of rock called shale. And they were first studied by this guy, a guy named Christian Pander. Any biologists out there may recognize him as being one of the founding fathers of the science of embryology. He's an accomplished biologist, an explorer, very interesting guy. But he also did a little bit of geology on the side. Those were the days when most people were polymaths in terms of their scientific aspirations. And Pander had seen some of these things preserved on the flat surfaces of shale. And this is a drawing from one of his mid-1800s publications. And what happened is when he would find these things, he would give them a species name. All organisms have a compound name made of a genus and species. In any given genus, nobody shares a species name, so that ends up being a unique binomial name. So originally, conodonts were all named as discrete pieces. And Pander looked at these and he decided these were all fish teeth. Now, what we're going to see is people put these in just about every category you could possibly imagine. We'll come to a few examples in a little bit. This is a picture of some of these conodont elements on one of these slabs of shale that's been split open. Sometimes we also fuse clusters. This is from some of my dissertation material. In all likelihood, what you're looking at here is the equivalent of an owl pellet. Some kind of predator gobbled up a couple of conodonts and pooped out the undigestible bits and pieces. But some of the pieces that people observed on shale partings that I've mentioned looked a little suspicious. Here you see a bunch of elements that are all laid out on a surface of the rock. And you can see the ones on the left side labeled with the letter PI on it, or P1. Those are called P elements. There are a whole bunch of different designations people use. I'm not going to get into that because there's so many different schemes. But the point of this is when you look at the distribution here, this looks suspiciously like these all came from one organism. It's not a random kind of clutter. You know, the big P elements up at the front are opposed to one another. They're smaller P elements immediately behind, and then there's those kind of longer elements in back. And this looked like something that might have had a soft body and had multiple conodont elements in it, and it died and sunk to the bottom. The body rotted away, and the elements didn't get disturbed, were buried in other sediment, and then were preserved in this nice linear fashion. And so what happened, the more of these kind of things people found, they came to realize that taxonomically we had a real problem. Conodonts are composed of multiple elements, sometimes paired, sometimes not, but each apparatus has multiple elements. What you're looking at here, this is from a genus called Promesium. This is from some shale in South Africa. These are extremely large elements compared to most conodonts, and this is a reconstruction of the apparatus. Interestingly enough, this material was restudied in the 1980s. That was when people concluded, hey, these were conodonts. Originally, they had been described as fossil aquatic plant material. As I said, they've literally been put in every category you can imagine. Plant, annelid worm jaws, you name it. Because if all you have are these disarticulate elements, it's kind of hard to figure out what the animal was if you don't have a body fossil. So people came to the conclusion that we had to deal with biologic species. And that makes things messy because when you name a species, if it's composed of multiple discrete elements, you got to go back and find the one that was named first. And that's the name you're going to use for the new organism. And all the other names basically get tossed out because they're now part of this new organism. The taxonomy is a mess. The closest analogy I can think of is if you only knew human beings from their teeth and you gave each different tooth a different name. And that happened for multiple decades, and later on, one day somebody discovered that all these teeth came out of the same set of jaws, you know. And you'd have a hard time reconstructing a human out of a molar or an incisor. So anyway, we developed these different apparatus. Now, the kind of holy grail, and I guess going back to the dragon kind of theme, I might want to think about Monty Python and the Holy Grail. There are a few dragons of sorts that appear in that for anyone old enough to remember that film. The Holy Grail for people who were studying conodonts was to find a body fossil. And we went through a few false alarms. One of the most famous ones, this is from work by Melton and Scott in the early 1970s. This is the Bear Gulch animal known as Lockria wellsi. The one up on the top is the fossil found in the raw rock. The one below is a photograph of the same sample underwater. And this is the artist's interpretation. And up at the top of in the center there, just above that little dark area, you can see some little things. Those represent conodont elements. And their interpretation was that these were some sort of filtering devices for food. And this had early on been proposed as a possible function of conodont elements. The problem is, it turns out you can't support enough soft tissue with filtering cilia on these elements and any of the reconstructed apparatus for it to function as a filter feeder. But early on, that was one of the things people considered. What happened, though, as they found more and more of these in the Bear Gulch limestone, people started looking closely at the elements, and they discovered it was just none of them had the same suite of elements in. And I imagine already there's a few light bulbs going off out there that are saying this was something that was eating conodonts or scavenging them. And that's essentially what this is. I mean, it's a legitimate organism, but it was not the conodont animal. And this was the lead article in a substantial volume on conodont zoology. So we've had some false starts, but what eventually happened in 1982, a British paleontologist was working with collections in a basement in one of the British Geologic Survey's buildings. This is a piece of rock from something called the Granton Shrimp Beds. And Ewan Clarkson had been interested in the Granton Shrimp Beds. These are some deposits that are located about 25 miles outside Edinburgh, Scotland, along the coast. They're called the Granton Shrimp Beds because they're full of shrimp fossils. And somewhere along the line in 1917, one of the individuals that was collecting material from the survey found some additional Granton material at another location that had soft-bodied preservation of organisms. You could actually see things like marine worms and jellyfish preserved in the rocks. And normally that kind of stuff all rots away. You don't have it in the fossil record. Well, Clarkson was aware of this. He was working desperately to try and relocate these deposits. And he also was looking at the samples that were being held in the British Museum from this 1917 collecting period. And one of the curators there hands him this slab of rock and says, I've always wondered what this thing is. And Clarkson immediately recognized that there were conodont elements down here at the front, and you can't see them in this individual photo. They found the original deposit that this was collected from. They collected another 10 or so body fossils of these organisms. They're very small, not big organisms. Remember, the conodont elements themselves, I said earlier, you might want to think of them as being about the size of a piece of large dandruff to small dandruff. Multiple things in the organisms. This is one of the other ones they collected. And in this lower picture, this bulbous area has been interpreted as having eyes. This is the head of the organism. And then the conodonts are located up up near the front. By the way, pronunciation, it's conodont or conodont, and I worked with these things, hearing both versions from different people I work with, so I'm going to slip back and forth, ignore it, pick out whichever one you like. This is their camera lucida interpretation of the first one, and the other fossils are much more detailed. But one of the things they note, aside from these big lobes at the front with the elements in them, is there generally is a hint of a nerve cord, a notochord going over most of the specimens longitudinally. Some of them have really nice V-shaped segments that look like they're muscle chevrons, the kind of V-shapes like you might see if you looked at a fish that you're going to have for dinner. Well, maybe before it was put on the dinner plate, but when it was getting cleaned. And then at the posterior end, there are usually some little lines that have been interpreted as representing fins. And their reconstruction after working with a number of these, this is one of the first ones that was done. And, this clearly is a nectonic, a swimming animal. It's something that's moving around in its environment. Again, relatively small. You can see that's 3 times magnification. I should have told you the elements that I showed you earlier from my dissertation were multiplied about 44 times, and each of those plates was just a regular 8.5 by 11 sheet of paper. So these things really are not very large. Other body fossils have been found, and these are just some other reconstructions. And I just chose this one to work with simply because I thought it looked reasonably nasty and intimidating. Now, what these things were actually doing in the environment is still very much open to debate. What you're looking at here, these are some coniform elements. These are usually really long-ranging species. They're not really good for dividing geologic time up. But one of the things to note here is they all have these kind of furrows going up the sides. And a Polish investigator in 2009 named Stanisiewski suggested that this might be the first example of a venomous organism in the fossil record. And what you're looking at here are, these are coniform elements that he imaged. And what you're looking at here are a bunch of modern vertebrates. A is from a venomous fish. B is from a venomous lizard. C, D, and E are both from snakes. And the idea here is whether passively or actively venom is injected into these furrows, travels down the furrow into whatever the organism is biting. And again, it's suggestive. We can't know for sure at this point that these things were venomous, but sooner or later we'll learn more about them. There are a whole lot of possibilities, but I think the idea that they were at this point active predators bears a lot of it's very favorable. Another possibility suggested by recent work involves this rather nasty looking guy. This is a picture of a marine lamprey. The mouth parts of lampreys are, well, they look like something out of a bad monster movie. They're full of a bunch of little tooth parts made out of keratin, similar material to our fingernails. And lampreys are parasites. This is a picture of a lamprey, pair of lampreys feeding off of a lake trout. These things got into the Great Lakes after the St. Lawrence Seaway was opened. They spend part of their lives in fresh water, then go back into the ocean, and then they devastated lake trout populations. What they do is they latch onto the animal. They use those nasty teeth to dig a hole in it, and they sit there and they suck juices out of the organism till the fish is ready to die, and then they have to go look for a new one to feed off of. Terrell and Henderson in 2018 suggested that lampreys might be a good model for conodonts. And you can see on the left is a lamprey with its mouth pulled out and the mouth parts highlighted. On the right, their reconstruction of a conodont apparatus. And one of the arguments against this has been that the conodon elements are made out of calcium phosphate, basically what our teeth are made out of. The elements on the lampreys are made out of keratin, which is basically a proteinaceous material. Well, Terrell and Henderson did some analysis of immature conodont elements, and in their interiors, and these things basically grew by adding successive layers of apatite, growing the elements in size. In the early layers, which eventually got covered by apatite, they found evidence of decay products of keratin. And so they've suggested that in fact, the lampreys and conodonts may be related. And this is something that had been suggested a long time ago. There's another similar organism called a slime eel or hagfish. That's a marine animal. It has the enviable lifestyle of burrowing into dead organisms on the seafloor and consuming them from the inside out. And slime eels, when they're disturbed, they exude an absolutely noxious sort of slime that drives off any predators that might want to consume them. But they have similar kind of mouthpart arrangements. At any rate, Terrell and Henderson suggested that that conodonts and the lampreys and the slime eels are closely related. Up on the upper left here, you can see a lamprey here. We've got the slime eels on the long bar there. And over here, we've got the lampreys in the dashed line. Right over here, you've got the conodonts ranging from the Cambrian up through the Triassic, and they share a common ancestor, or would in this proposed cladogram would share a common ancestor. Now, I've done a bit of stretching to somehow relate these little things to dragons. And I will say in my defense, if you look at this, on the lower part of the diagram, we see a bunch of primitive fish and other things that we're pretty comfortable thinking of as vertebrates. And you can see that they're ultimately end up being joined to the same, somehow there's a common ancestor between them and the group that led to the conodonts. Again, this is all suggestive work at this point. So that's going to be my defense of the title Ancestor of Dragons. I know I've been overly liberal, but let me talk about some fun stuff before we get to my dull, boring work, the stuff I like to do. You can do a lot of interesting things with conodont elements. This is a picture from a demonstration against above-ground nuclear testing during the Kennedy administration. You can see a lot of women there. Women were very active in the attempt to limit above-ground testing. And part of that was because one of the forms of fallout generated by above-ground tests is a radioactive strontium isotope that has a real short half-life. It breaks down very rapidly. And when it breaks down, it will shoot out sometimes what's called an alpha particle, 2 neutrons and a proton. And the concern here is that strontium, because of the arrangement of its outer shell of electrons and its size, it can easily substitute for calcium. And the thought is, if I'm a mom and I'm given or a dad for that matter, I'm giving my baby a bottle of milk, and that milk came from a cow that was eating grass that had fallout on it that included radioactive strontium, some of that strontium might get incorporated in my child's bones and at some point may decay. And when it does that, some of those particles that are emitted might go through a cell nucleus, might go through a gene and cause a mutation, leading to cancer. And it's a legitimate concern. Now, conodont elements incorporate strontium. If we look at seawater, the ratio of strontium 86 to 87, which have extremely, I'm trying to remember, 86 I think is stable. The point is, both of those, they're the same element. 87 has one more neutron in it than 86. It's a slightly more massive atom. They stay in seawater. An individual strontium atom will stay in seawater for about 25 million years. Water in the ocean stays in the ocean on the order of a few thousand years. What that essentially means is that at any given time period, the ratio of strontium-86 to 87 in the ocean is going to be the same, no matter where you sample the ocean water. 87 is mostly derived from the weathering of granitic rocks on the continents. A lot of 86 comes from basaltic rocks forming at the mid-ocean ridges. So Burke, back in 1982, decided he was going to investigate these ratios over the entire span of the Phanerozoic. So you see back here, you see the beginning of the Cambrian over here. And then on the far left side of the diagram, we're essentially up to the present date. And you see the scattering of lines there. Now, what he did was he used other researchers' work. They would use hard parts, calcite shells of animals called brachiopods. They would use whole bits of limestone, because what happens? Sometimes an animal with a shell, it's grabbing calcium out of the water to build its shell out of calcite, calcium carbonate. Every now and then, it would grab a strontium atom instead. And so it would be strontium carbonate incorporated. And what happened when it would make a mistake, it could care less whether it was strontium 86 or 87. So if there was more 86 in the water at a given time than 87, the shell would have a little more 86 than 87, and vice versa. And Burke knew that these ratios had changed over time. So he basically comes up with this plot. Now, in fact, this has been much more refined. I mean, you see a wide scattering, although on the left side, for modern times, where the sampling's much better, it's pretty close. One of the things I did in my dissertation work is in southeastern Wisconsin and in upper Michigan and in the Door Peninsula there, I did have a couple spots where I had biostratigraphic control, where I could say that the rocks represented a specific time period. And so I had the same time period in the south based on the conodonts as I had up in Door County and upper Michigan. And what I did was I had these samples run for strontium. Fortunately, the conical elements, they have an affinity for making mistakes. They incorporate a lot of strontium in their system, but there doesn't seem to be a preference one way or the other for 86 or 87. And they are the most common conodonts you find. I sometimes, some samples I have cooked literally hundreds of pounds of rock from a single unit to get 3 or 4 elements that were biostratigraphically significant. So I had two levels in southeastern Wisconsin, 2 horizons in northeastern Wisconsin and upper Michigan, where I knew where they fit on the geologic time scale. And I ran strontium levels on them. And essentially what happened, using a more recent, although this is relatively dated, what I did was I plotted strontium. You see the strontium ratios up on the top, and nowadays we go another 2 decimal points over for the ratios up there, but that was the limit at the time, was 4 decimal points. I plotted my samples. They're all the little things you see with the K's there, and the little lines up above the diamonds represent error bars. When I plotted the ratios for the samples that I knew which conodont zones they came from, they plotted exactly where they should have. And once I knew that worked, I could take samples from areas where all I had was these coniform elements that didn't tell me anything. And I could interpolate, I could use their strontium ratios, and generally they all fit in exactly where they were supposed to. There were a few exceptions, but this was actually, I believe, the first time anyone had done this with Silurian age rocks, and I probably should have long ago gotten off my butt and published it, but the bright side is it's been my dissertation has been much quoted, so it hasn't sat on the shelves worthlessly. Now, this is a recent article I was involved with publishing, and you can see me there. I'm kind of halfway down the line from the lead author. That's primarily because all I did was the Conodont work on this. Wesleyan, if you look closely, Wesleyan actually gets a little publicity here. It has North Carolina Wesleyan College as my place, so I've done a service for the school. This article involves attempting to correlate between the interior of the Michigan Basin and the exterior in part. What you're looking at here, you can see the outline of Michigan there, right up in the upper left corner. You can see a location map, so that's the part of the United States we're in. This pink band of material is inside the Michigan Basin, this area that's slowly subsiding 400 some million years ago. All the little dots there are what are called pinnacle reefs. They're just small reefs that kept growing upwards. As the basin subsided, the corals kept growing up and stayed up near the surface. That whole pink thing was actually surrounded by a huge barrier reef system, not unlike the Great Barrier Reef in Australia. And it enclosed this entire basin. And it's very problematic to correlate rocks from inside the basin to outside because they're very different environments. To give you a better feel, this is from some supplemental material in the article. This gives you a little idea where North America was geographically during the Silurian. You can see here it's sitting well south of the equator and north of 30 degrees south. It's basically we're in the tropics. So this was an area, limestone, corals, they only form in tropical environments nowadays between 30 degrees north and 30 degrees south. So one of the things they did to try and work on correlating in and out of the basin is to use carbon isotope ratios. And all I'm going to tell you about carbon isotope ratios is we usually use something called the delta carbon 13 ratio. Two kinds of carbon you worry about in this. There's carbon 13 and carbon 12. Again, the only difference is one has an extra neutron. Carbon 12 is preferentially taken up by organisms. And we have a variety of ways of measuring the ratio of these two carbon isotopes in rocks. You can do this with just literally a piece of limestone, because the calcite, there will be little bits of carbon, right? The calcium carbonate, right? The carbon from the mineral is there. Some of that will be carbon 12, some of it will be carbon 13. And the calcite doesn't care which one it is. It's whatever gets together out of the seawater or the organ. The limestone basically is made of bits and pieces of hard parts of calcite organisms that get broken up. So whatever they've been grabbing out of the seawater. And this stuff, just like the strontium, has a long enough residence time that it's the same everywhere in the ocean. So what you see here, this is a worldwide chart of excursions in carbon isotopes. There's a standard that everything is measured against called the PD belemnite. A belemnite was kind of a squid-like organization around in the Cretaceous period. Some of you may recognize PD. You may have heard of the PD River in South Carolina. This belemnite was pulled out of some rocks along the PD River. So that's the standard that we use for evaluating carbon isotope ratios. So these are excursions either above or below the ratio for that guy. And I'm just going to focus on a lower part of this so we can expand it a little bit. These excursions, we can find them everywhere in the world as long as we have continuous rock record. And a lot of them have been named. What we're looking at here is the Iraquivian excursion. And you notice maybe some familiar names there. You see right here we've got Pterospathodus, Amorphonothoides, Amorphonothoides. Well, what a friggin' mouthful. The last appearance datum, that's the end. That guy doesn't show up anymore. So if you have that, you know you're in older rock than anything above. A little above that, you can see first appearance datum, of Cocolella walliseri. I talked about that earlier. That was the guy that came from this guy called Cocolella ranuliformis, which you see down here. And you also see Ozarkidina sagitta renana, which coexisted with ranuliformis, but disappeared before walliseri shows up. So if you find ranuliformis and sagitta renana together, you know you're below the walliseri zone. And essentially, my part in that article was to recover conodonts from well over 100 cores. And these are just some visible light photos of these elements. And we're just going to look at a few of them up close. This is Cocolella ranuliformis. All the little tooth-like things on the top are referred to as denticles. This is Cocolella walliseri, and the only difference between it and ranuliformis is you can see up near the 7, up at the top, you've got a little lateral process here with a few denticles on it. Here's another picture, and this below is a lateral picture where you can see this process. And so these samples, walliseri was recovered from just above samples that had ranuliformis in it. Pterospathodus amorphinathoides, the other one I talked about, was recovered lower down, and this was the last one recovered. Everything above it, it was absent from. And the thing that allowed us to do was to look at sections within the basin. This is from the well that I worked with, and I'm going to do a close-up of this in a minute. This section here represents similar rocks in southeastern Wisconsin, including stuff from that quarry we looked at earlier. So here's a close-up of this, and you can see the biozones here. The significant thing here is I've got Cocolella walliseri up here, and none of the samples above the top of that pointer contained Ozarkidina sagitta renana or ranuliformis. So we know this is literally a pinpoint in geologic time. And the reason that's important is if you look on the right side of that part of the diagram, you see the carbon isotope curves. But one carbon isotope curve looks just like another one. Some of them are a little bigger excursions than others, but they don't all show up with the same profile everywhere in the world. All you end up with is a bunch of squiggly lines, and you need something to peg these squiggles so you know that this particular squiggle is the Urakavian excursion. And that's essentially what my finding these conodonts allowed us to do. And so we could go from inside the basin to southeastern Wisconsin, well outside the basin, where I have walliseri and ranuliformis again. And you can go to other parts of the world. We can find the same sequence of conodonts in Great Britain, and we can correlate these rocks as being of exactly the same time period. And once you peg one of these excursions, you don't have to worry about the ones up and down, unless you've had a period where the rocks were eroded away. As long as you've got a continuous rock record, you can name those excursions as you go along. So above the Urakavian, we have the Maldi excursion. Don't have to do any conodont biostratigraphy. Once we've got that lower one pegged, the next one up has to be the Maldi, and so on. So it actually is something that has a lot of utility in terms of dating. One last thing about conodonts, and I will stop subjecting you to this. One of the interesting things about them is that when you heat them, they change color. And this is work based on work done by a woman named Anita Harris. She was Epstein at the time. In the early 1970s, she was working in the Appalachian Mountains, and she was collecting conodonts as she walked from the eastern side of the mountains across all the way down to the slopes on the western side. And she started to notice that the conodonts were different colors. And she found that in the center of the Appalachians, they were one color, and then as she proceeded west, the colors would change. Likewise, if she was in the center and proceeded east, the colors would also change, and they would be in the same order. They were distributed basically symmetrically. And what she realized is that little bits of carbon that was preserved in the conodont elements, if you heated them, and these are laboratory-induced color changes, you could tell the temperature of the rock. And this led to the creation of something called the Conodont Alteration Index. And I'm not particularly good with this thing, but you have different what are called CAI volume values. And people can look at these and they can tell roughly how hot the rock got. And the importance of that is that oil companies are looking for rock that hasn't been heated too much. If you have a lot of carbon in a rock unit, you need that to make oil. But to turn it into oil, you have to heat it up enough to start breaking up some of these long carbon chains. If you heat it too much, it turns into natural gas, and most of the time it's floated away. And Anita Harris gave a talk about this at a Geologic Society of America meeting, and she literally, when she left that meeting, got mobbed by representatives of all the major oil companies because they realized that they could use this to save themselves a lot of money. If they were drilling an exploratory oil well in an area and they could find these bugs, they could determine how hot the rock got. And if it never got hot enough to generate oil, that's it. They're not going to do any other wells in that area. You know, if they found the rocks were heated to just the right temperature, even if the first exploratory well they drilled was dry, they might still invest in looking around in some of the surrounding area. Or likewise, if the things got too hot, you know, that they wouldn't look further. So this is one of these areas where, you know, I like these things just because I enjoy the process of finding them, but it actually has some economic utility. It saved oil companies a lot of money over the years, which I can't say is necessarily the most socially desirable thing, but it certainly is economically. All right, well, I'm going to leave off there. Yeah, I'm guessing I've probably overextended my welcome. Anybody have any questions? Well, all right, that's it. You're free to go or to stay for the great debate, which I don't think I quite ran into. It's only 8:17. Thank you. Have a good evening. Yeah. And very sorry about the lack of a reception, but I just didn't think it was prudent to do given the current virus situation. Sorry I went so long. Oh, that's right, I got to get out of the way here.
At this time, I wanted to recognize present and past Jefferson Pilot Award professors. If you're in the audience, please stand. Dr. Leverett T. Smith, Jr., Dr. Douglas J. Goodwin, Dr. Richard L. Watson III, Dr. Kenneth Finney, Dr. David Jones, Dr. Linda Flowers, Dr. Paul J. DiGategno, Dr. Alan Johnson, Dr. C. Dale Theron, Dr. Mary Lou Steed, Dr. Barbara L. Perry Sheldon, Dr. Marshall Brooks, Mr. Everett Edelman, right there in the audience, Dr. Jay Quinnen, Dr. Steve Farabee, Dr. Vivian Anderson, Dr. Fred Grissom, Dr. Erica Kozel, Dr. Ron Smith, Dr. Jay Stubblefield, Dr. Nancy Floyd, Dr. Bill Yankowski, Dr. Jonathan Saris, Dr. Carol Lawrence, Mr. Roger Drake. Dr. Kimberly Martin, Dr. Mayor McGall, Dr. Fred Sanborn, Ms. Kathy Winslow, Dr. Heather Louch, Dr. John Temple, and Dr. Lee Templeton.
Good evening. The 2020-2021 Jefferson Pilot Professor has worked for North Carolina Wesleyan College for 16 years. His record of scholarship includes 2 published books, winning the Pinion Press Novella Contest, publishing numerous book reviews and encyclopedia entries on contemporary authors. This professor is currently working on a 2nd novella and a 2-act play. His contributions to North Carolina Wesleyan College are also significant. Since arriving to the college, he has served as program coordinator for journalism and faculty advisor to the decree. In 2005, 2006, he was selected as Advisor of the Year by the SGA. Unless you have lived under a rock, see what I did there, Dr. Kuglitsch? You know we are talking about Dr. Bill Grattan. Dr. Grattan has also served as the coordinator for the 4th Monday Faculty Colloquium, co-director of the Writing Center, faculty meeting secretary, and has also overseen 9 honors projects. Dr. Grattan has also served his community through his work with St. Francis Church, Stop Hunger Now, WUNCFM, and the North Carolina Media Advisors. Through his teaching, he has demonstrated his skill at drawing students into thoughtful, critical discussions about course-related issues. He is constantly thinking about his effectiveness as a teacher and exploring ways to improve his teaching abilities, of which qualities to ensure that the students will always receive the highest quality instruction. Please help me in congratulating Dr. Bill Grattan as the 2020-2021 Jefferson Pilot Professor. And I'll end by saying the Jefferson Pilot Committee, who's made up of past Jefferson Pilot professors, is the one who makes recommendations to the president, who then selects the final recipient. And this is typically kept a secret until the night it's revealed, which is tonight. And it was especially hard to keep the secret since this has been delayed by several months. So now it can be stated, and we congratulate him and give him a good pat on the back. So congratulations. Thank you, Dr. Kuglitsch. Thank you guys all for coming.